Hello friends, welcome back to my channel Agritech once again. Please do not forget to like and share my channel. And I also would like to request you all to subscribe my channel. Please do follow my videos and please help me to promote my channel as well. And today's session, I'm going to describe about genetic engineering. I'm going to give you general information about what is genetic engineering and what is its history. And I was thinking about to give you the information about methods and basic steps involved in genetic engineering, but I came to realize it's gonna be overwhelming for you and it's gonna have a lot of information. It may not be digestible for you. So I'm splitting the content, and in today's video, I'm going to describe about the definition and history of genetic engineering. Then in next video, I'm going to give you the methods and basic steps. So let's proceed towards the definition. What is the genetic engineering? What comes in your mind if you think about engineering? Maybe you think like making something else, making some building, making some structure, isn't it? Likewise, in molecular science, what we do is we reconstruct or we construct some genetic material within certain bacterial cell or certain biological cells or in any biological cells using its genetic material. This is what exactly is genetic engineering, and I will give you more like illustration from one of the example from Gibson assembly cloning. First, what we do is we make a like desired fragment of DNA that we like aim to transfer into the another DNA material. So for example, here is insert one maybe from the one DNA and insert two from the another DNA, but they are the specific fragment of DNA from some genetic material. And what we have linear vector, another vector. It has different set of genetic material. And next, what we do is, you know, we insert these like what we do is we insert these foreign DNA to the linear vector in a specific way. Then finally, what happens is we will have a recombinant DNA which had a different set of genetic material now, like a hybrid, we had linear vector that had different set of genetic material. Now we have inserted some DNA from genetic material from different DNA, insert one and insert two. Now the recombinant DNA will have three different set of genetic material, and this is actually the example of Gibson assembly cloning. And this is also called a like recombinant DNA. So the transformed DNA will have different set of genetic material, different set of genetic information. This is what genetic engineering is. The better DNA have constructed, transformed, and it have a new set of genetic material now. And let me illustrate from one more example. What we have here is we have a chromosome, and we gonna transform that yellow section. It's a desired fragment of our DNA. We have to use restriction enzyme. You can follow my previous video where I have given actually about the restriction enzyme, how it works and why we need restriction enzymes. We need restriction enzyme to cut the DNA. So in this example, what we're gonna do, we're gonna like transform the genetic material from one chromosome. We use the restriction enzyme. Then we have a vector. We also use the restriction enzyme to open up that vector. Then that desired fragment of DNA gonna insert into the complementary ends of the, like once we open the vector, it will have the complementary ends. Then we're gonna insert our desired desired fragment of DNA. Then what will happen? We will have the new set of genetic material, including both kind of genetic material from vector and from the donor DNA as well. And DNA ligase is is an enzyme that helps to stick the ends from complementary ends of vector and the desired fragment DNA ends. So it will help to link all genetic material and make a new set of genetic material there. It's a genetic engineering. There are different way to manipulate, different way to edit gene. There are different methods called gene slicing, gene cutting, and recombinant DNA technology. Recombinant DNA technology of genetic engineering is highly used and successful examples. It's being practiced over the world, so it comes always in the spotlight. But there are some other way to do the manipulation, like gene silencing and gene splicing. And genetic engineering is one of the useful method to develop some vaccines, some treatment, and it's it's being used in everywhere, in agriculture, in pharmaceuticals, in in medicine as well. um, do you know, for the immunological treatment, scientists use genetic engineering to develop vaccines. And in some pharmaceutical studies, scientists use genetic engineering tool to develop medicines, antibiotics. And in agriculture, what genetic engineering have importance in is we can develop adaptive plants, adaptive animals, high yielding plants, animals through the genetic engineering. We can make the genetically modified organism that might have the a different set of genetic material, and that result into giving some sort of new content, new taste, new flavor, new vitamins, new minerals in a single food. That's why genetic engineering has all important importance in any kind of science, like food science, agricultural science, pharmaceuticals, and maybe in immunological. And another one important fields of study within genetic engineering is immunology and cancer. There are scientists who are working hard through the genetic engineering approach to develop different therapy to cure cancer as well. So let's proceed with the history of this important applied science, applied approach and method in science. So isn't it is so interesting, like people have identified genetic engineering, now it's getting a blessing to everyone. Now I would like to proceed with history, and I have developed some like state, or I would like to say what is, I have developed the history into the 4 or 5 different sections. First 1960s, 70s, 80s, 90s, 20 and 2010. Maybe 6 different category, or six different era. And I'm going to give you the information about what happened in that era especially. Before going to the 1960s, I would like to give you information about 1950s, what happened. During the era of 1950s, Watson and Crick have developed a model of DNA. And at the same time, at the same era, scientists have identified, or no, they have discovered, they have prepared synthetic DNA in test tube. It was so interesting, like since 1950s, this genetic engineering been came into the spotlight. But I found that since 1960s, the genetic engineering actually came in spotlight. I mean, it had been started from 1960s to grow up as a genetic engineering. So at the era of 1960s, what happened is the scientists have identified DNA can be caught, and it can link by using the ligase. So at the 1962, scientists have identified green fluorescence protein. It comes from the jellyfish, and this green fluorescence protein is commonly used to identify the location of certain organelles, or for the different other experiments. So it has one of the importance in fluorescence microscopy, where we can find the targeted gene or targeted organelles. And in 1967, scientists have identified the DNA ligase. The ligase enzyme, it helps to link the cord fragments of DNA. So that was the one of the important discovery during this era. And in 1968, at the late 19 late 1960s, at 1968, scientists have identified the restriction enzymes. Restriction enzymes, you can follow my previous video. What are the restriction enzymes? They cut the desired fragment of DNA into different sections, and so that we can manipulate DNA, isn't it? So it was the one of the wonderful era of genetic engineering, where they have identified, scientists have identified the ligase. I mean, they have identified DNA, the caught DNA. DNA can be caught and it can be linked together by using ligase enzyme. So during the 1970s, what happened is the genetic engineering was lifted a bit than 1960s. Scientists have identified these type second restriction enzyme, for example, Hind second, BAM, BAM as I second kind of enzymes. They have purified these enzymes, and at the same era, the scientists have identified the gene splicing experiment to develop the recombinant DNA. That time, they have identified actually to how they can cut the DNA, and they can make a recombinant DNA. And that time, it was called splicing. It's called the gene splicing method. And second type, like type second restricted enzyme used for mapping of DNA. And scientists have identified one restriction enzyme, type second restriction enzyme. Maybe that was Hind. I'm not sure. Maybe it was Hind second restriction enzyme. Scientists have identified that it can cut one of the viral genome into the 11 different fragments. It was one of the key discoveries, so that we came to realize like DNA can be caught and it can be manipulated. And the one of the important discovery during the 1970s is like the scientists have identified, like created recombinant DNA by cutting one of the DNA from donor DNA, and they have manipulated that DNA into the vector DNA and made a recombinant DNA. And National Academy of Moratorium, they have started like studying genetic engineering experiments. And one of the important like discovery in this era is development of hybridoma technology. What happened in our body is human body have actually have beta cells, we call B cells, and they can like replicate themselves and produce monoclonal antibody. They can clone itself and produce monoclonal antibodies. So scientists have started working on it, and they were trying to mimic the property of beta cells. And they have merged the plasma membrane cells. Let me find a exact like name. It's a myeloma cells. These are the cancer plasma cells. They have like scientists have merged these cells to the beta cells, and they were trying to find the monoclonal antibodies from there, so that they can treat the immunological, they can like use in a treatment, immunological treatment and some diagnostics. So they got success at the like era of 1970. It was 1975, the hybridoma technology first came in spotlight, and they have successfully develop monoclonal antibodies by merging the beta cells with cancer plasma cells. Isn't it so interesting? And during the era of 1980s, first time the vaccines and treatments to the human been approved. They all been like developed through the genetic engineering. And one of the important discovery at this era is to development of synthetic insulin that used to treat diabetic patients. And another one is development of polymerase chain reaction. The polymerase chain reaction that help to like always amplify, that help to amplify the segments of DNA. So we will be able to like see the if the fragment like the DNA is present in that material. There are different kinds of machine used to see the amplified DNA segments. First we should have the PCR machine, and another one is like seeing under the busy, like making it visible under the gel, different gel, agar gel, and other gel as well. So the PCR, nowadays during this COVID crisis, scientists are working hard to develop vaccine, and in simultaneously what in different countries, the COVID-19 is like testing through PCR, polymerase chain reaction. At this like with PCR, what scientists or technician do is they take a specific fragment of um, like desired DNA, then they do amplify within certain like a catalog, a certain recipe, then we can see amplified DNA under the certain gel, agar gel or something else. Isn't it so interesting? They have identified the PCR technology at 1983, and we are still using PCR. So that was the one of the revolutionary development through genetic engineering. And at the same era, 1980s, the development of zinc finger nuclease been discovered, like it's developed. This GFN helped to like target the specific gene. And another one is the first recombinant vaccine for human been approved at the 1980s. That was the vaccine for hepatitis B. And first time BT corn appears on the field at 1988. At the late 1980s, scientists have already worked to make genetically modified corn, and that had already been appears on the field. So interesting, isn't it? The next one is the era of 1990s. Era of 1990 was a blessing for discoveries of cloning and GMOs. At that era, scientists were working to like find how they can clone and how they can make a genetically modified organism to the plant and animal both. And important discoveries were like discovery of principle of CRISPR. It's a gene editing tool. I will give you the more insights on next sessions what is CRISPR, but it is the one of the gene editing tool. It's using widely, uh, and it's a cluster regularly interspace palindromic sequence is called, and it has Cas9 protein, and which act as a scissor to cut DNA in some specific sequence, and it's being widely using these days. But yeah, I have told you I'm going to give you the more information in the next video, where I'm going to give you what are the genetic engineering methods. This is just a history. And another one is they have like cloned dolly sheep first, sorry, yes, they have discovered the CRISPR first, and then next they have discovered the dolly sheep. This is the first huge animal being cloned ever from a one adult cell with the same genetic material. The sheep has been cloned and it's named Dolly. That was the one of the revolutionary discovery that time. Scientists have got success to clone a huge animal as well. And at the same era, the first time the human chromosome being sequenced as well. And let's move on to the 20s. What happened during the 20s? And during the 20s, the scientists have got success to find first gene-targeted drug therapy. And that was for the cancer, that was called the chronic myelogenous treatment. And it's for the chronic myelogenous treatment. It's for the cancer, one of the cancer. And first time in history, the glowfish, which had a fluorescence protein, and these kind of fish been sold as a pet. And another one is FDA approval of first preventive cancer vaccine to human, and another one is first introduced the IPCS at 20s, and important discovery at 20 as the human genome got sequenced, and new regulations like ethical regulations regarding the cloning and some other sequencing thing came in the spotlight. Scientists like realized like using like cloning animals may not be beneficial in a criminological and some other like animal right perspective. So the new regulation been set and human genome got sequenced at 2020s. Let's go on to the what happened at the late 20s. Now it's almost describe about today's generation, like what happened during 2020s, after 20s, you know. The first synthetic life form developed, you know, scientists got success to develop a cell. This is the Mycoplasma mycoides. They have developed a synthetic cell using like they, and that cell had like genetic material and live cell they developed in lab. That Mycoplasma mycoides hasn't born. It had been developed synthetically. And another like gene editing tool, TALEN, that had been like discovered at that like that period, you know. And another one is the discovery of CRISPR. I mean, they have discovered how CRISPR can be utilized. And I have already told you it has Cas9 protein, and it has a specific ability to cut or DNA sequence which has palindromic sequence. I will give you more insight in my next video. And another one is identifying the possibility of gene drive. You know, if there is a gene drive, there might have some bias to like inherit the genetic material from parents to their offspring. And scientists have identified the possibility of gene drive at 2014. And another one is like first treatment like therapy for cancer been approved for genetic engineering. This is called CAR-T therapy. And first GMO, you know, salmon being sold in market and human embryo edited with CRISPR, and first human trial for CRISPR approved. It's mean late 20s or within after 20s to this is 2020. This era like heavily flourished with finding the treatment for cancer, finding treatment for like some sort of vaccines through the CRISPR and some other gene editing tool. And they have developed some therapies for cancers. And you know, and these days scientists are also working to find the vaccine for COVID-19, and they are using some sort of gene editing tool. Is I have some understanding, scientists are using the recombinant DNA technology and some other like gene editing tools such as CRISPR and other TALEN technology to identify or to develop the vaccine against the COVID-19. And you know, our history, it's been developed from 1950s to till 2020, and still we have huge scope of genetic engineering. Manipulation of genetic makeup is working in a beneficial way to treat human and animal disease, you know. So it has a very huge um, it has a very like huge impact on human and animal and plus in agricultural like productivity, human like cancer treatment in human and finding some antibiotics. So knowing the history, we I would like to conclude that genetic engineering is one of the two approach. It's never gonna end. It's always have some sort of space to explore itself, and always would be like beneficial to treat some of the uncurable diseases through the medicine, such as cancer, diabetes. So I would like to ask a question. How did you find my uh, today's presentation? Whatever you like the most during today's presentation, mention below on the comment. And thank you for watching my video. We'll come up with the methods and the basic steps involved in genetic engineering in my next video. So till then, have a good day and enjoy your day. Stay healthy and stay safe at all time.